In this case, I think I'm going to call to mind in the supplementary acknowledgments the lessons learned from 5 conferences that we attended this year. And I want to give an acknowledgement to Nature magazine, which last year published a series called 8 Meetings That Changed the World. So what I'm going to start by putting on the blackboard is something that might be called 5 meetings that are going to change the world. And I'm going to review for you in language a little more vivid than I put into this supplement exactly what those meetings were and what themes were covered and why I view these as potentially world-changing meetings. Not world-changing for the results that were presented there, but world-changing because of the attitudes and the tools that were presented there. So, a conference that was known as SQUINT. So, Seattle Quantum Information Theory Conference. Worst acronym ever in some respects. And I guess the I is capitalized, SQUINT. And the theme of SQUINT is that, now I'll put a nice little color code on here, is that quantum system engineering is a practical reality. Now what do I mean by quantum systems engineering? And what do I mean by is now a practical reality? Well, systems engineering is defined famously by Simon Ramo as the design of the whole as contrasted with the design of the parts. So quantum systems engineering is no different. It's the design of quantum systems, the prediction of their behavior, the optimization of their function, considered as a whole as contrasted with considered as the amalgamation of their parts. And the nice thing about SQUINT was that people were doing this with all kinds of systems, mechanical systems, trapped ion systems, optical systems, and there was an emerging sense that not really very much is mysterious about quantum systems, whether it's considered from a theoretical point of view or from an experimental point of view or from a simulation-based point of view. It's not clear what we can do yet with quantum systems. That's the design aspect of it. But taken component by component, we've developed a very good understanding and a very solid confidence that when it comes to quantum systems, we know what we're doing. Now the SQUINT meeting was followed by the FOMS meeting. And FOMS is Foundations of Molecular Modeling and Simulation. That took place at, I don't even know how to pronounce it, Semi-Mono or Semi-Yahoo. I had to look it up in order to get it spelled right in the syllabus. There, people were articulating heresies. That heresy, yes here, biological structures are generically dynamic. And the person who was articulating that heresy was David Shaw with his Anton computer. He was showing animations to the wonderment of all, extending over an entire millisecond, which, given that individual fluctuations occur on a time scale of a femtosecond, is a very, very long time. And the dynamical behavior revealed was so conformationally variable as to really call into question how much biology crystal structure alone can tell us. And this set the stage for the second heresy, which was articulated by the UW's own David Baker, which was that when it comes to dynamical structure, that simulation of dynamical structure, it's not perfectly reliable, but it's is or can be more reliable than what has been for 2 generations the gold standard crystal structure. Now, both of these sound a little bit heretical, but they aren't really heretical. You know, if you think about how biological organisms must actually work, that virtually every element of a biological organism has to be dynamical, has to be radically active in its conformation. And given the self-selective nature of crystallography, that you can only crystallize things that aren't very dynamical, it's necessarily the case that reliance on crystallography restricts your view of the mechanisms of biology to those mechanisms that are not hugely conformational. So these Fomm's heresies really aren't heresies at all about things that we thought were true but aren't. They're a kind of a different kind of heresy. They're the heresy that maybe the tools we have conditioned us to look in not the wrong places, but not to look in all the places that we need to seek for biological truth. So those say that already sophisticated experiments in magnetic resonance require fairly detailed quantum simulations. Some of these experiments run for weeks and require rather intricate sample preparations. And pulse sequences that are not at all the result of experimentation, but that are the product of system-level design. And there's greater and greater reliance within the magnetic resonance community on software simulations that predict in advance not so much the results of your experiment, but the general parameters of the experiment that you're about to run. In other words, for molecules of the scale of the molecules that you're interested in. And let's say structural features whose details are not known but whose size is generically known. Will such and such a pulse sequence reliably give you the information that you seek? And there are a host of programs, including spin evolution and Simpson, both referenced in the extended syllabus, that use density matrix theories to get at that answer. Now, at the same time that the simulations are coming up in capability, the sensing technologies are coming down in capability. Dan Rugar and Dan Weidekamp were both at the ENC, and they both showed experiments involving magnetic resonance signals originating from a number of spins that is large but countable. Signals from ensembles on the order of, say, 100 to 1,000 spins. And there is a trend that has been existing throughout the history of magnetic resonance to build instruments that are more and more sensitive and are sensitive to smaller and smaller numbers of spins. So it's pretty clear to everybody at the 50th ENC that those two capabilities were about to cross over. That we are approaching a world in which we can simulate every spin in the sample that we're observing. And this is, means that rather radical changes are coming for magnetic resonance. That magnetic resonance will start to resemble more and more Navier-Stokes fluid mechanics, in which you can make a credible attempt to simulate any system that you might feasibly hope to build. And you'll find in that supplementary material kind of a benchmark, which is a cubic nanometer of water. There are about 65 spins in that cubic nanometer of water. That's too many to be simulated by existing packages because the dimensionality of the Hilbert space is 2 to the 65, which is about 10 to the 20. But the theme of this seminar is that in the real world of magnetic resonance, things are sufficiently noisy that you don't really need to use that whole state space. You can use what we call a concentration and pullback strategy, and you can start to hope to simulate in full quantum mechanical detail every spin in that cubic nanometer. Now, a cubic nanometer is chosen partly because that would be 65 spins would be a pretty significant extension of existing quantum simulation capability. It would be simultaneously a pretty radical extension of the quantum, so it would be an extension of simulation capability, it would be an extension of microscopy capability. But also, nanometer resolution is the resolution that the FOMS people tell us they need in order for their dynamical simulations to start taking over. So this isn't just a twofold convergence in spin simulation capability and sensing capability. It's a threefold convergence with nanometer scale microscopy. So what we're really talking about in this seminar is a comprehensive overlap of the FOMS goals, the Kavli goals, and the ENC goals. Now, science is great, but and engineering is fantastic, but at the end of the day, you really have to start deploying your science and your mathematics and your engineering in service of real-world problems. And that brings us to the fifth in our series of conferences, which was the American Chemical Society in DC. In particular, their amyloid structure and protein assembly seminars. And here I have specifically in mind so what these diseases have in common is not that they're caused by viruses or genes or toxins, but they're really conformational disorders. Emphasize how very naturally the themes of these conferences then tie together. The people at Squint believe that everything is quantum, and of course they're right. And that follows that every dynamical process is a quantum process. Every communication channel is a quantum communication channel. And one of the themes of our article is that you can stop right there. You don't have to consider things like noise processes and thermodynamic processes because those are equivalent to communication channels and control channels. So the language that our manuscript develops is that all of quantum mechanics boils down to symplectic processes and metric processes. There's a real sense in which that's not even a new result. All we've done is taken together results that are solidly established and are in the quantum mechanical literature and written them down in the symplectic and metric language. That symplectic and metric language comes straight from FOMS because molecular dynamical simulations already use symplectic and metric language. So really we're going to set out to deliberately blur the boundaries between classical and quantum simulation. In fact, we're going to present, just by dialing an index in our simulation, we'll be able to switch back from fully classical to fully quantum or anything in between. The point of view that we're going to develop then is that the microscopy goals of the Kavli conference are best understood in this symplectic and metric language that the quantum computing community and the molecular dynamics community already share. So these communities don't just share similar goals of understanding biology. They share a similar natural language, the symplectic and metric language of mathematics. All of that is provided partly as motivation for what is going to be the second half of this morning's talk, where we start wading into exactly what it means to have a symplectic state space and for trajectories to preserve symplectic structure, and what it means to have a metric state space and for trajectories to preserve metric structure. So with that in mind, we're now going to take a 5-minute break.